All right, it's noon. Noticing that that clock is 3 minutes slow, but my phone says it's noon, so we'll start promptly according to digital time. Um, Thank you all for coming. This will be our last Center for Evolution and Medicine seminar for the fall 2019 semester. In the spring, again, we'll have a smattering of invited speakers and current faculty and current trainees giving presentations. So please, if you're not on the listserv, you can go to the Center for Evolution and Medicine website and sign up for the listserv to get announcements about the seminars that we're having. They're also live streamed, and I think we'll continue to be live streamed in the spring. And so if you can't attend in person, you can attend digitally. Open your chips if you choose to open them now. And so with that, I'll introduce our last speaker for the semester. Chris Cotsapas is coming to us from Yale, and you can go find his CV and bio sketch, and it's all very impressive, and he's done many wonderful things. And I'm super excited to have him here because I met him at an NIH Office of the Director, Office of Women's Health Research, day-long workshop on incorporating sex as a biological variable. So you may or may not know that in late 2014, it was announced officially 2015, the National Institute of Health says future research must consider sex as a biological variable. There's been very little enforcement of that, I would say, and actually some published papers that there's been very little enforcement of that. And I don't think that it's all malicious. I think a lot of people have been doing research in a certain way for a long time and not thinking about the effects of sex chromosomes or sex differences. And the pipelines and the pathways that we have don't typically incorporate them. And so Chris chaired this meeting, and I think in a couple ways it really left a big impression on me because one, he made sure that everyone got a chance to talk, and that doesn't always happen when you have really famous and smart people together in a room. Some of them will dominate, and especially the one that's chairing may, of course, has many good ideas. Yeah, pontificate, has many good ideas, but the meeting wasn't like that. It wasn't just listening to one person. So it was really great. We got the opportunity to hear from everyone that was there that wanted to talk. And we talked about a whole slew of things. So from generating sequence to actually even recruiting your cohorts to aligning the sequences to doing sex stratified analyses and trying to figure out where we're going from there. And I was just so excited about it that I immediately emailed everyone in the department and said, we have to get you here. And we did. And so thankfully, it wasn't too difficult to entice him to Arizona from Boston in December. So without taking up any more of your time, I'll let you go. But thank you so much for coming.
Thank you, Melissa. Wow, thank you. Thank you very much for the invitation. Thank you all for coming. I know it's the end of the semester. You have a notable absence of snow. You should get some. We have 6 inches. We'd be happy to send as much as you want. So I've been thinking, I've been basically hanging around conferences, hanging around conference bars, asking people 2 questions for the last few years. One is, what's the most important thing that you're not working on? And the other is, why do you think more women than men get autoimmune disease? And I get really interesting responses to both. And the reason that I've done this is partly sociological and partly because I'm probably having the first in a series of midlife crises where I don't know what to work on next. I want to do something important. I want to work on something interesting. There are so many glaring problems out there when you start asking questions. And this was a way for me to sort of sniff around and try and figure out what I was working on and what I should be doing. So at this point, I was going to tell you that I have a confession to make, that I want to manipulate you. That the reason this is basically an hour-long ad and a sort of an attempt at manipulation because I want expect you to all sort of think about sex more and sort of work on sex more, but I'm preaching to the choir in this case. But nonetheless, the point of, I think, this seminar for me is to organize my thoughts around what the sex disparities in autoimmune disease are specifically, and how this could be a template for thinking about sex and human traits more generally. And I think it is instructive enough that it's worth wasting an hour of your cumulative time to sort of go through some of this stuff, because there are subtleties that I think a lot of people don't appreciate necessarily that are disease-specific, and there are subtleties that are technical, I think, that we don't talk about enough. So I'm hoping that you can teach me something about this. So the autoimmune diseases are a weird, heterogeneous mix of somewhere between 80 and 100 distinct clinical entities, depending on who you talk to. Most of them are complex traits. Some of them are clearly Mendelian, but the vast majority are complex. There's no nice simple inheritance, but they are heritable. There's some debate in the literature. People use the word autoimmune to mean certain things. People use the word inflammatory to mean certain things. Autoimmune generally means the presence of antibodies, and antibodies directed against self-antigens, and inflammatory just means inflammation. They are two sides of the same coin. I'm not super sort of keen on that distinction. I think it's somewhat artificial. But basically what happens is your immune system decides it doesn't like some compartment of the body. And if it doesn't like the GI tract, you wind up with inflammatory bowel disease, or in some cases celiac disease, for very, very different reasons. If it doesn't like the skin, you get scleroderma or you get psoriasis. If it doesn't like the brain, it punches holes in it and you get multiple sclerosis, which happens to be something that I work on. If it doesn't like the skeletal system or the muscular or the joints, you get rheumatoid arthritis or you get ankylosing spondylitis. If it doesn't like the more sort of general endocrine system, you get something like lupus, or you get Sjogren's disease, which is an attack on the salivary and lymph glands in the face and neck. And if it doesn't like your metabolic system, for instance, especially the pancreas, you get type 1 diabetes. Type 1 diabetes is the type that kids get, where they get insulin injections. It's not grandma's diabetes, it's the childhood diabetes. So these diseases are all fairly common, in that common's kind of a plastic word. It doesn't mean 40% of the population. It means somewhere 1 in 1,000 and more frequent. So it's not specialist clinic type of stuff. Rare disease tends to be like the 1 in 100,000 syndromic things that we talk about, often named after dead white guys, increasingly now renamed pathologically, because that's actually sensible and it tells you what the disease is, instead of having to remember what, I don't know, Barrett's esophagus is. Turns out that Barrett was a guy who had an esophagus. He doesn't have one anymore. So these diseases are also heritable. How heritable they are, kind of up for grabs, but you see familial aggregation, you see clear evidence of genetics. You also see that they are not Mendelian. There is not one mutation that segregates in a family, and if you inherit that mutation, you have the disease, and if you don't, you don't. That's just not how these diseases work. And so over the last 15 years or so, we as a community, and we as me and my lab specifically, have done a huge number of increasingly large, increasingly elaborate genetic studies trying to identify the genetic determinants of these diseases. So this is kind of what I cut my teeth on as a postdoc. I did some of the first associations in rheumatoid arthritis and lupus, in what are laughably small sample sizes these days, but it was impressive way back then. And sort of going forward, most recently, if you look up at this sort of Circos plot that looks impressive but is virtually impossible to read, in multiple sclerosis, when we looked at a cumulative total of maybe 125,000 individuals, we now know there's at least 200 loci in the genome that drive common variant associations. And there's another handful that drive rare variant associations. And these two things don't seem to overlap. So the common variants, genome-wide, explain something like 20% of the heritability, and the rare variants explain something like 5%. The disease is on the order of 60% heritable. We haven't explained the other 35% of that heritability. We're not quite sure what's going on now. This is just the heritability, that which is attributed to genetic factors. So we're not sure what's going on. Some of this is sample size. Some of it may be overestimates of heritability, and so on. But this is fairly typical. This has also been true if you look over here. Here's rheumatoid arthritis that has an equivalent sort of picture. And here's IBD, again, similar sort of picture. And when you look at other autoimmune diseases, the same kind of idea. Very polygenic, complex, pretty heritable. Gwas works. We've got a lot of these loci in the genome. And now one of the questions has been, increasingly over the last 10 years, what do these tell us, right? Genetics is a game of desperation. It always has been. The only reason you do genetics, really, is you're trying to understand the biology of the trait you're studying, and everything else you have done has failed. Because it's not the obvious way to do it. The obvious way to do it is to actually study the biology of what you're doing. And actually, we've made remarkable progress in studying the biology directly of a lot of diseases. We've also made remarkable lack of progress. In particular, one of the things that it's been extremely hard to do is separate correlation from causation. When you look at a bunch of patients, how can you tell what is epiphenomenon of disease and what is causal? How do you know that the causal factors even still exist when you're looking at people 20 years after the onset of disease? Sounds like obvious, but actually it's a pretty big problem. And genetics offers hope that maybe if you look at the genetic factors that are driving it, you will arrive at the causal biology of disease. And so, way back when, the idea was, great, we do a GWAS, we'll find a variant, it'll land in a gene, we'll figure out what that gene does, and boom, like it's 15, 20 years of work, but you'll get the causal biology. And then biology happened to us, because biology is like an onion. It has many layers, and every layer makes you cry. And what happens is that it turns out that the heritability is not concentrated in gene bodies, but generally concentrated in regulatory elements. We showed this like a really long time ago with John Stamm, this big project that John Stamm did as part of the Roadmap Epigenome Mapping Consortium, where they looked at DNA's hypersensitive sites across a whole bunch of tissues. And then what you see is that there's enrichment of disease-associated SNPs in DHS and DNA's hypersensitive sites that are accessible in tissues relevant to those diseases. And that's true in the immune cells, that's true in metabolic disease, it's true all over. More formal analyses subsequently have sort of really confirmed this. So if you partition heritability across the genome into different compartments, the ones that always light up are DHS specifically, but more generally things like enhancers, K27 acetyl-marked regions, and so on. And so it looks like King and Wilson were really right in 1975 that most change that is phenotypically relevant is regulatory rather than gene coding. Which sets up a problem, really, which is, okay, how do I find the regulator? They're usually small stretches of DNA. They're not as easy to find, certainly sequence-wise, as genes are, because genes are constrained. Actual coding sequence is heavily constrained. There's a codon structure. You can find genes. It's actually quite hard to find things that are predicted to be marked as K27 acetyl enhancers, for instance, right, just from sequence. So you've got to do the experiment. And then how do I figure out what these things do? We don't have any tools. We have very few tools. If someone gives you an enhancer out in the middle of nowhere, trying to figure out what tissue it operates in and what genes it controls and under what circumstances is actually pretty difficult. You can do it for one, but there's millions of these things. And there's lots of associations to lots of traits. So how do you scale this? And if anyone says CRISPR, I'm leaving. right? That's not the answer. That doesn't scale yet. And so you need to work a little bit harder. And so we did. So one of the things you can do is you can start from GWAS, and you can basically project the posterior probabilities of association onto regulatory elements. Don't worry about the math too much. It's not that interesting. But you can start with your posterior probabilities now, project them down. These are the variants. This is, if the more yellow it is, the more posterior probability it has of driving association. You can project these onto regulatory elements and then ask, how do these connect to genes? And then I can partition the probability that this is driving risk. I can partition that up between the genes that it regulates, right? And then I can do the same for this guy, and then I can get the partial probability from here and the partial probability from here, add them up, and now here's a probability that this gene actually drives the phenotype. This, of course, is the difficulty. And so we did something pretty silly, right? Sort of really, really simple. You just say, I've got an atlas of 100 cell types in the Roadmap Epigenome project where I've got paired expression and DNase hypersensitivity data. Okay. Let me correlate for every DHS, let me correlate whether it's accessible or inaccessible in a tissue with the expression level of or nearby genes, and get a correlation coefficient and use that to split up this. Because so far, I'm just talking about genome, right? I'm talking about physical position on the genome. I understand that. This part is hard. And so we did that, and it turns out it works pretty well. It took a postdoc in my lab who is now an assistant professor at Western University in Canada, like 4 years to figure this out. I kind of helped, like I popped in occasionally and told her about what colors I thought she should use for the plots. But so here's like a classic example, right? Sort of this is the CD58 locus in multiple sclerosis. This has been known to be an association. We know that CD58 is the gene for a given value of no. It's reasonably good. The complication is that there's this other gene called CD2, which is its interacting partner. These are two surface receptors. They're expressed on different cell types, and they mediate the interaction between T cells and antigen-presenting cells. So when you look at this locus, it could be CD58, could be CD2, could be either of them. And you're unmoored. These are now regulatory regions. So there's no reason that just because the signal's over CD58, it couldn't be a regulator that drives CD2, because enhancers act at distance. And that's the problem, right? So it turns out that most of the posterior probability lands on a single hypersensitive site, and that is strongly correlated to CD58. And it really looks like CD58's the gene. And here's the expression evidence. In blue is the expression of CD58 in tissues where that mark is not accessible, and in orange is where it is accessible. There's this really strong correlation between them, and that's why we think this guy regulates this gene. As a random other gene, VANGL1, here's the same DHS, here's the pattern of expression of VANGL1 when that's not accessible to when it's accessible, and there's no correlation. So it's really unlikely that that element has anything to do with this gene. Make sense? So you can play this game and you can map a whole bunch of genes, and we're proceeding now. Now we have a general system where we can say this enhancer drives this gene, and I think it's quite likely that this is the risk. And here's now the mechanism. So we're following a bunch of these things up, and that's where it gets a little harder. But at least now we have a rational system for generating hypotheses. Now, of course, if you've got a regulatory element that controls a gene, you would expect that one of the things that happens is that the expression of the gene changes. It could be contextual, but it could just be just the expression level. In which case, this is an EQTL, right? The gene expression itself is a quantitative trait because its level is being set by a genetic variant. And we're interested in that genetic variant because it's associated to disease. And so you should be able to take a Gwas trace, this is a locus on chromosome 12, and find that the gene expression trait, when you do a Gwas for gene expression instead of MS, it should look really, really similar, because these two things are actually driven by the same underlying effect, right? And this is actually the expression of a gene, metal21B, the one in red up there, in CD4 T cells and in CD14 monocytes. And when you look at the data, it's really clear that there's strong correlation between basically the p-values of association of one or the other. So we actually designed a sort of formal statistical method to co-localize them. It's joint likelihood mapping. So you're asking, what is the likelihood that SNP1 drives association in trait 1 and SNP2 drives association in trait 2? And you do that for all pairs of comparisons of markers across that region. And you either have likelihood maximized on the diagonal, so it's the same variant that drives both, or it's maximized off the diagonal. And that's the test. And then you say, okay, these two look like whatever the effect is, it's the same, or it's not the same. The good thing is you don't have to know what the underlying marker is, and that's good because fine mapping doesn't really work yet. Okay? What's really surprising is if you go through a whole bunch of autoimmune diseases, look at CD4 T cells, CD14 monocytes, and lymphoblastoid cell lines as a weird B cell representative, there are a lot of loci that have an expression trait that looks right. It's nearby, but actually very few of them, only 37 of 261, actually look like the expression level is driven by whatever is driving risk to disease. And that's because there's a lot of expression QTLs all over the genome. So anywhere you look that has a gene, you'll find an expression QTL, just that most of them are irrelevant to the disease association you're looking for. So that's a little disappointing. So this is kind of the state of play right now in autoimmune disease and probably more generally in complex traits in humans. And so the question is, how do you get from these to mechanism? And I think we're pootling on and designing sets of experiments, thinking about that. But at the same time, another big problem is this sex bias in disease. And as we've gotten at least the basic ideas of genetics, now that we think we know how to do complex trait genetics, then maybe it's time to use what we've learned to start looking at sex biases in disease. And so what I want to do for the remainder of the talk is try and convince you why that's important and why we've been ignoring it, and maybe a little bit of what we're going to do over the next few years. So the reason that I put a Vitruvian woman here is because when you look at the rates of autoimmune and inflammatory disease across pretty much most of the population, they are really, really biased towards women. Globally, across all 80 or 100 entities, it's something like 80% of diagnoses are in women. That rate varies from something like 13 to 1 female to male bias in things like Sjogren's disease, maybe 9 to 1 in lupus, 10 to 1, all the way to 3 male to 1 female in something like ankylosing spondylitis and primary sclerosing cholangitis. Some diseases are not biased or not significantly so, but a lot of them are, and they're not biased 60-40. It's 80-20, it's 90-10, and so on. It's a really, really strong bias. And the short answer is we have no idea why this is. And this is what I learned over 5 years of drinking with people at conferences. At most, you get some idea of, someone actually told me this in not too dissimilar a tone. Well, women are designed to tolerate allografts, Which is a complicated way of saying pregnancy, which is one of the stupidest things I've ever heard. But, I didn't think very much of the person to begin with. I thought even less of him later. One of the interesting things, when you dive into this, it gets a little bit more complicated. So it's not just that the diagnoses are biased. The sex bias is increasing over time, for instance. This is data for MS, and this is the sex bias in diagnosis rate. So this is twofold higher in women than men, going up to threefold. So it's 1.5 to threefold. And what you see in different cohorts is that generally the bias increases over time as you march into the present day. And there are several reasons for this. One is access to healthcare. MS is a notoriously vague disease to diagnose and was until the advent of MRI, where you could actually see holes in people's brains, which happened in the 90s. And before that, the actual symptomatology is quite diffuse. People have tingles and peripheral numbness. They have episodes where they lose balance, they feel unwell, and then they recover because the disease usually relapses and remits, right? And it turns out that those relapsing remitting cycles correlate to new lesions appearing in the brain, and then the brain is relatively plastic, so you kind of wire around it and compensate, and you kind of get better until you don't. Okay, but it's actually quite hard to diagnose, and quite frankly, I would guess that in the 1950s, if a man came in and said, you know, I've got peripheral numbness and I don't feel well and sometimes I lose my balance, you know, their doctor would say, okay, sort of, let's look at this. And if a woman came in, they just, in the unlikely reason, in a likely situation where they came in, they'd just be told to go home and stop complaining, right? So there is access to healthcare bias here. However, every time the criteria for disease have changed, the bias has gone up. And the criteria have always changed to include milder disease. And you actually see this over time. One of the few really objective measures you have to look at progress or progression of MS is the time it takes someone to get in a wheelchair, right? There are two components to MS. There's the physical component where people have trouble ambulating and eventually they use a cane and eventually they're in a wheelchair, they can no longer walk. Usually that correlates to a lesion on the brainstem, but not always. And the other one's cognitive. And cognitive testing is still not routine, but it is becoming more routine. But most of the tests have been physical. And the one thing that you can really, really sort of basically hang your hat on is when someone's in a wheelchair and you know that they've reached that milestone. There's not a lot of resilience to that. There's not a lot of, I feel better, I feel worse. When people are in a wheelchair, they're generally in a wheelchair when they've gotten there. And so one way that people routinely measure progression in the MS field is how long has it taken you to get into a wheelchair? And this is time to wheelchair in men and women. And men generally get into a wheelchair faster than women do, by a period of 6 months over a course of, 35 years. It's not a lot, but it's definitely there, and it's been replicated over and over again. You also see more lesions in men. You see generally higher disability. You see more cognitive decline in men with MS. There are fewer of them, but it's fewer, right? So there's more to the sex bias than just incidence. The disease actually seems to have different courses. And that's not only true in MS. So if you look at sex bias, one of the things that you see is that most of these diseases occur in adults. Stereotypically, when you talk about autoimmune disease, they are diseases of women of childbearing years. That's how the literature has described them for years. And it's true. It's women in their 20s, 30s, 40s. Some of them are older. If you look at the early onset diseases, there's no sex bias. The stereotypical one is type 1 diabetes that occurs in childhood, and it's 50-50. If you look at late onset type 1 diabetes, it is much more prevalent in men than women, by about 3 to 1. If you look at the diseases that occur after puberty, MS is pretty biased. As you get to menopause, you start seeing something more interesting. You see progression and burnout in MS around menopause. And you get a 50-50 split. About 50% of people go on to this secondary progressive phase where they just get steadily worse, massive cognitive decline, sort of very serious problems, and the disease pretty much kills them. And then you get another 50% where they stop getting new lesions. They stop having this inflammatory insult. There's not a lot of new lesion activity. There's not a lot of anything. You generally take them off meds. They have accumulated burden and disability, but the disease becomes inactive, and that's called burnout. If you look at MS that occurs in very early childhood, or MS-like things, again, there's no sex bias. If you look at the diseases that occur around menopause, like lupus, SLE is systemic lupus erythematosus, generally just called lupus, or rheumatoid arthritis, again, highly biased between women and men. So, what are the differences between men and women that could explain this? And this, I think, is where everyone gets stuck, including us. How do you start thinking about differences between men and women? Because they're pretty profound. Right? So there are some pretty obvious differences, right? One is the allograft hypothesis. We have a whole bunch of rather powerful steroid hormones washing around various systems, and they're different between men and women. We actually have a slightly different genome. And more generally, it could be that the associations that we've seen on the autosomes are sex-specific. It could be that the risk factors that we're mapping in these Gwas's actually have differential effects in men and women. Families of hypotheses. You could also talk about X chromosome inactivation here, or skew in some way. So those are the obvious things that people have talked about. So let's start with the easiest possible one. So we'll start with the genetic effects being different in men and women. So this has been shown really nicely by Cecilia Lindgren, primarily, amongst many others, but Cecilia's work in anthropometrics has really shown this clearly, where if you look at men and women, this trait is waist-hip ratio. It's a measure of adiposity. So your waist and your hips, it's basically body shape. It's where you put fat. And men tend to be apple-shaped, and some women have pear shapes and some women have apple shapes. And it turns out that this central obesity, this apple shape, is correlated to cardiovascular disease. So when you adjust for BMI, you can actually map determinants of how you store fat in your body, whether it's peripheral or central. And the central obesity is the bad one. Men generally have central obesity. Some women have central, some women have peripheral. But you can see that a lot of effects are different between men and women. These are GWAS hits in autosomes. They're not on the X chromosome, but they have different effect sizes between men and women. And all the way up at the top, you have things that are effectively neutral in men. They do not affect the trait at all, but have a strong effect in women. And down here, you have things that are very disparate that have opposite effects. So this allele actually increases waist-hip ratio in men and decreases in women. We have no idea what's going on. But it is possible, at least in human traits, to do that. These analyses have largely not been done in autoimmune disease. In the cases where we've looked at, we've been doing it in MS. There's no such clear pattern. There's maybe a tiny amount of evidence for sex-specific associations. We've got 200 of them, but there's no overwhelming observation. So when you separate men and women, instead of putting all cases and all controls together, you do a fully stratified analysis. And this is where the technical stuff becomes important. Because it turns out, of course, that 80% of our cases are women. So we don't have enough power to look for associations in men because we don't have enough male cases. And when I say we don't have enough, in the rare variant association study that we did, we looked at 38,000 cases and 34,000 cases and 38,000 controls. Out of those 34,000 cases, only about 9,000 were men. So when I'm doing a 25,000 versus 9,000 association study, I don't have power to detect differential effects. I'm looking for difference in differences now, right? This is a second order problem. This is an interaction effect. I can't see that. What we need is something like 50,000 and 50,000. So one of the things that we really have to do is actually rethink how we put these cohorts together. And one of the reasons I think we haven't made that much progress, especially in the autoimmune diseases, is because they are so biased. We don't have the raw material to do this. So we're going to have to figure out how to do that. And that, I think, is on the agenda for the next 5 years. And as weird as it sounds, one of the things we're going to have to do is go out and recruit more men. We have a lot of women. It's the men we don't have. But what this implies to me is that the liability threshold may be different in men and women. If you think about it, if this is true, then what it means is you need more alleles to get men over that hump into disease than women, either because there's a protective factor or because these alleles just don't have the same magnitude of effect. So what do you expect? Here's the overall risk, and in women it's kind of more smeared out. And here's this notional line where if you cross this line, you get disease. In women, it's kind of smeared out, and so you get more people actually crossing that threshold. In men, for whatever reason, it's either pushed down this way or the distribution's narrower. And so less of the population crosses that magical barrier. What this means is they're probably outliers, and you would also expect them to have more severe disease, if that's true. So that actually kind of fits with what we're talking about, right? Where you see more severe disease in individuals. Either that, or another way to think about this is this threshold. They both look that way, but this threshold's different. The nice thing about this is typical stat gen, right? This is like the classic modeling overall risk in the population. So you can do this at bulk. You don't have to look at individual associations where you don't have power, because you don't have enough men or you don't have enough women. You can do this across the whole genome in bulk and actually ask the question. So liability threshold modeling may be one way around our sample size limitation. And that's something that we're starting to follow up on. But we don't have an answer. We genuinely don't know. It doesn't look like the genetics are different. But what I can tell you is that if you look at the distribution of risk burden in men and women, so if you just sum up how many risk alleles any individual carries and multiply them by the effect size to get the total risk burden, these are controls. These are cases: celiac disease, IBD, multiple sclerosis, rheumatoid arthritis, lupus, type 1 diabetes. MS, RA, and lupus are the three strongly female-biased diseases here. Type 1's not really biased. IBD's maybe a tiny bit biased. Celiac disease isn't really biased either. Male and female risk, and you can see that it's largely purple. So the distribution of risk overall in the sexes is not different. And that suggests that men and women are not differentially biased. It's not as if men have a lot more risk. And so that's maybe an argument against this liability threshold thing. So whatever's going on, certainly it's not enough difference in risk between men and women, because remember, these are ascertained, right? These should be purple and these should be starkly different blue and red. 80-20, 80-20, 90-10. This is not a subtle ascertainment. If men needed a lot more oomph to get over that line, you'd expect them to have a lot more burden. And there are many problems with this analysis. This is like a really sort of quick and dirty. For one, it relies on effect sizes and on risk alleles that were found in Gwas that are female biased. And so maybe that's a problem. But certainly we're not seeing anything super obvious here. And you would expect it with a degree of ascertainment. If we were talking about 60-40, you could lose it in the noise. But 80-20 splits between men and women and 90-10, I would have expected to see something. So I don't know. On the other hand, this is a really interesting story from Steve McCarroll's lab. So Steve found the association of the MHC alleles to schizophrenia. And when they dug into it really carefully, they found that it was a copy number variant in one of the complement genes right at the edge of the MHC that was driving that. And it seems to have something to do with synaptic pruning. Following this on, they kind of looked at lupus and asked, okay, well, is the MHC association in lupus due to these complement genes instead of the classic HLA DQB1 genes that people have always thought about in lupus? And it turns out that that's part of the story. I won't go into all of this, but basically, if you look at the complement genes up at the top, if you have the short version of the what they're calling the B haplotype, so it's an expansion, but you have the short version of the expansion in the C4b gene, your risk goes up hugely. What's interesting is that it is very differential in men and in women. The risk is mostly in men, not in women, by a factor of 3. And we have no idea why. And again, people have been working on the MHC since the 50s. The serotype associations with autoimmune disease have been there since the 50s. The specific types have been there since the 70s. And this is the first we've heard of this in lupus. I have no idea what this means. I can barely get my head around this. But there's certainly new things to find. And there are sex-specific associations to find. Whether this is an exception or not, I have no idea. But I think it's part of the story. So in MS, if we start thinking about the sex chromosomes specifically, we did a whole bunch of linkage in the 90s and early 2000s. One thing we did not find is really strong associations on the X chromosome. Here's the linkage trace for chromosome X. That's 1. It's exactly what you expect. That's the best you can do. In GWAS, we find 2 loci on the X chromosome. Type 1 diabetes, which is not biased, has found 1 on the X chromosome. There's a couple in lupus. As far as we can see, in terms of GWAS, the X chromosome's kind of behaving like a normal autosome. When you adjust for gene density, it's got the expected number of loci, risk loci in it. There's nothing massive that could drive this 80-20 or 90-10 split, those extreme splits. There are caveats in that the analysis of the X chromosome is complicated. Very often, genotyping the X is a little bit more complicated. You guys know this. I'm not going to go through all of this, but it's, again, you would have expected that at least in linkage, something that was driving a sort of 5-to-1 split between the sexes would have come up in linkage, and it's not coming up. And this was in 700 and something families. This was not like 10 families, right? This was a big linkage study. You might have seen something. X chromosome inactivation also trivially does not seem to be the culprit. This is a paper from Tanuja Chitnis, Peter Gregerson, a whole bunch of people. When you look at normal individuals, these are all women. Normal individuals, here's the skew of X inactivation in peripheral lymphocytes. And here's the skew that you see in lupus, type 1 diabetes, MS, and juvenile rheumatoid arthritis. There's nothing terribly obvious with the degree of skew. It looks the same as you might expect in normal individuals, which is kind of all over the place. There's clonality in the leukocytes, so it's not a 1-to-1. It gets a bit complicated. It's fairly coarse, but it's not obvious. This has been an idea that's been floating around that magically X inactivation explains this skew because the paternal allele is antigenic, and so you will tend to raise autoantibodies or self-reactivity to that allele that is differentially inactivated in different compartments of the body. This seems to argue against it. Again, the jury's always out, but right now that's not apparently an answer. Moving on to the sex steroid hormones in pregnancy. Here's the onset risk in years for MS. And it increases basically from puberty going onwards. Here's what happens during pregnancy in women who already have MS. This is the rate of relapses in the population. And you see that it goes down during pregnancy. The biggest decrease is in the third trimester. You really get a substantial suppression of relapse rates. And it pings right back up in the first trimester postpartum. And what appears to be happening is whatever is happening in pregnancy is dampening down the immune response. And that's kind of consistent with fetal tolerance. The other thing it's consistent with is anti-inflammatory effects, not so much fetal tolerance, but generalized anti-inflammatory effects of things like estriol that's produced by the placenta. And as soon as you withdraw that, you've basically saved up the relapses, and they happen immediately after postpartum. So after birth, when there's no longer this dampening, it comes right back. So you're not, at most you're delaying it by a few months. You're going to get that relapse, right? And this kind of makes sense. So Rhonda Voskuil at UCLA did this really interesting clinical study where they gave glatiramer acetate, which is a standard therapy in MS, with or without estriol in women. Estriol group is red, placebo is blue. Again, all of these women are receiving glatiramer acetate that's either supplemented with estriol or not supplemented with it. And estriol actually makes the drug more effective. It decreases the number of relapses and the time to relapse. Sorry, it increases the time to relapse that you see on the therapy. Estradiol does not. It's specific to estriol. No idea why. Interesting, though. I don't know what would happen if you gave men estriol. I'm not aware of any data on that. It's an interesting idea. I'm not sure it's ethical, but it's interesting. I think you are inducing gender changes without, or you're inducing secondary effects that will be known. I don't know. Doesn't it?
Well, I mean, but it doesn't in a female-specific way.
True. I don't know. It's interesting. So, speaking of sex hormones, we tend to think about menarche and menopause as being important determinants of autoimmune disease. And here's one of the reasons. If you look at menopause, this is in the Nurses' Health Study. Lupus, and one of the reasons that this is done in lupus is actually pretty common. And so if you're looking in even a series of 100, 150,000 women, you'll get enough cases. If you're looking at MS, lupus is 1 to 2% of the population. MS is 1 in 1,000. You just don't get enough cases in an observational cohort to do any subgroup analysis, ever. So in lupus, the probability of premenopausal versus postmenopausal incidence is skewed. You're likelier to see incidence of the disease after menopause. If you look at whether women have used perimenopausal hormones, you see a pretty strong effect where hormone use correlates to increased risk. These are all perimenopausal hormones, very specifically. There's no real effect with oral birth control. Age at menopause certainly seems to have a trend where earlier menopause is an increased risk factor, but not the total number of ovulatory years. It's something about age at menopause, not whether that sort of is a sort of shorter reproductive lifespan. If you look at postmenopausal women, basically what you're seeing is that the disease actually is not as severe after menopause. So if you look at women before, and if you look at women who express the disease before menopause, it attenuates after menopause. And if you look at women who are diagnosed after menopause, the disease is weaker or is less severe than women who were diagnosed before menopause. So there's something about menopause that attenuates lupus, and probably RA as well. On the other side of the spectrum, in MS, here's pubertal timing in men with risk of disease. This is time of puberty. It goes from about age 8 to age 20. This is the density of data. As you expect, most boys experience puberty somewhere between 10 and, say, 15. And there's no effect on risk of MS. And if you look in women, earlier menarche is a risk factor. Very early menarche seems to be a risk factor for MS, and that's actually reasonably well known. We don't really understand why, but more recently, Brent Richards and Steve Saucer did a Mendelian randomization experiment because the genetics of menarche have been mapped out to a certain extent. There's something like 400 associations now known. The genetics of MS are reasonably well known. So you can play games like, do variants that increase age at menarche affect MS? Is there a causal relationship here, which is what you would expect? And the answer is, yes, it does. If you look right up there, that first line, the odds ratio for MS per yearly increase in age at menarche goes down. So early menarche is associated with higher risk of MS, right? As soon as you adjust for BMI, that goes away. Because it turns out that one of the correlates of age at menarche is BMI. High BMI induces earlier menarche. And that's a pretty strong association. If you separate these SNPs into BMI-related SNPs, so they increase puberty and they lower BMI, they're associated to risk of MS, and the ones that aren't are not. And this is not just association, this is a causal inference argument. So it's not even just menarche, it's something more complicated, it's something about BMI. But it gets worse, right? So this is now the effect of BMI on menarche. And I won't take you through all of this, but basically there is an association of BMI, of menarche on BMI that goes away once you adjust for BMI at age 8. So whatever it is that is driving the age at menarche is also driving BMI at age 8, way before menarche. So something is happening that actually has effects on puberty, on pubertal timing, that is also driving BMI. And this thing, whatever this thing is, appears to have a knock-on effect eventually on the immune system. And my brain's already fried. I don't know about you guys. I have real trouble kind of thinking through this. I can do 2 variables. 3 variables start becoming difficult. So as far as I can tell, this is mostly where the field is. So what do? There's all of this disparate research. There's all of these clues. It's not as if people are stupid or ignoring this. Like, people have taken runs at these problems, and we keep coming up with these tantalizing clues, but we don't know what it is. So puberty would have been such a nice story, or right? So, or reproductive lifespan or something like that, because then you can just drive it right to probably the sex hormones, and then Bob's your uncle. And that doesn't seem to be what's going on. At least not the whole story. Because it turns out that when you look at sex steroid hormones, they have a profound effect on the immune system. This isn't perhaps a new story, but this is some data from my lab. So these are naive T cells, CD4 T cells, isolated and exposed to very, very low levels of TNF-alpha. And what you're looking at is NF-kappa B signaling, which should result when you expose T cells to TNF-alpha, right? Immunology 101. It took us 2.5 years to teach ourselves how to do this, but it's really simple immunology. I'm not smart. So here's what happens at vehicle, and here's the response you get to TNF, right? This is the degree of phosphorylation of P65, which is one of the NF-kappa B subunits. This is a classic way of measuring NF-kappa B signaling, right? And it goes up, exactly what you expect. Here's what happens when you pre-expose those T cells to estrogen, estradiol. It actually goes down a little bit, right? So the net level of phosphorylation is down a little bit. And here's what happens when you expose them to TNF. The signaling is blunted by a lot. It's about 60, 70%, depending on how you're looking at it. This is one sample. We've done this in like 20 individuals, and it's generally true. It's just a little bit more easy to access when you look at a single individual. What's really interesting is this reverse dose effect. So this is increasing amounts of estradiol, and you actually get a decreasing influence on signaling as you increase the estradiol. I have no idea what this means. We're not even sure that this is real. We see it about half the time. We don't see it always. So we're not sure if it's real. But something's happening here. The blunting effect is generally true. Whether you get this reverse dosage effect, I think the jury's still out. I want to believe, and Irina is much more skeptical than I am, so she sort of puts me in my place. But expression in general does seem to be different, right? Again, here's another story. These are a whole bunch of B-cell relevant genes expressed in skin of men and women from keratinocytes or from healthy skin in, let me see if I can get this right, because I really need to get this right. That's healthy human skin up there. This is a transcription factor that controls them called VGLL3. That's in skin, and that's in isolated keratinocytes. But what becomes really interesting is when you look at VGLL3 distribution in healthy skin, in women, it's in the nucleus, and it's doing something. And in men, it's not. It's cytoplasmic. When you look at cutaneous lupus samples, it goes into the nucleus in women as well, in men too. So there is a switch here where this transcription factor, for some reason, is largely inactive and expressed at low levels in men. And once you get, it's my last one. And once you get disease, once you get lupus, you switch to a female phenotype. No idea what this means either. So what are we going to do? We have to do a whole bunch of stuff, I think. I think one of the things we do is we actually start thinking genetically. We can overlap things. We have a way to declare whether associations are the same. We can use shared heritability. So we can go through and build what we're causing causal chains, right? So you take a GWAS of MS, and then you take a GWAS of hormone levels. You actually go out and do a GWAS. So we're trying to do this in Finland and Sweden now, and try and collect maybe several thousand individuals, circulating sex hormone levels, and actually do a genetic study. The UK Biobank also has data, and overlap the associations, and actually find things that are driven, where risk is driven by the same allele that is also driving corticosteroid levels, if such things exist. You can look at other cellular phenotypes, which we already have. We've mapped a whole bunch of these things, and we're working on them. And then you can also add expression levels here. And you build up this picture where the same allele is driving changes to the expression of gene X. It's driving changes to cellular phenotype Y. It's associated with changes to corticosteroid Z, and that results in MS risk. And then you start playing games like Mendelian randomization to show causality, but that's how I think you're going to get to mechanism to a large extent. That's one of the things that you need to do. This is going to kill me if I don't wrap up, so I'm just going to end with my acknowledgements. Cecilia Lindgren in particular has been like a huge influence on why I started doing this. Shamil and I have been working on methods for years. With John, we've been working on a lot of the regulatory regions. Ingrid and Thomas are very old friends and colleagues in Sweden who we've been doing MS epidemiology, and we've also been doing a lot of the sex steroid hormone work. Mark is my postdoc mentor who still actually remarkably takes an interest and helps me. He's in Finland these days, and so we've been setting up corticosteroid GWASs in the entire Finnish population with Mark. And Jakob is a colleague in Denmark who's also gotten us access to the entire hospital register system in Denmark where we can do that. Whole bunch of people in my lab, some of whom are still there, some of whom have gone on to bigger and better things. Speaking of my lab, come work with us. It's going to be a fun 10 years. Thank you.